Start with the basics in dermatology. First, let's do the skin. Skin skin weighs 4 to 5 kgs of our total body weight. It acquires 1.7 meters square of the area in total. Now let's move on to the layers of the skin. It has three main layers: epidermis, dermis, and subcutaneous fat. Now further, epidermis is divided into five layers. First is stratum corneum, which is keratinized and anucleate. Second is stratum lucidum, which is translucent, and the translucency it is provided by the LED granules, and it is present in palms and soles only. Stratum granulosum is the next layer, which contains keratohyalin and lamellar granules. Keratohyalin is basophilic in nature and it contains filaggrin. And this filaggrin mutation is responsible for atopic dermatitis and ichthyosis vulgaris. This lamellar granules in stratum granulosum is frequently asked questions in the exams. These are also known as woodland bodies, membrane containing granules, woodland granules, cementosomes, etc. This provides barrier function to the skin through lipid. Next layer is stratum spinosum, which is also known as prickle cell layer, and it is rich in desmosomes. Last layer is stratum basal, which is also known as stratum germinativum. Then let's move on to layers of dermis. There are two layers of dermis. One is papillary, which is 1/10, and another is reticular, which is 9/10 of the dermis. And then it contains hypodermis or subcutaneous tissue, which has two compartments, septa and lobules, and inflammation of this subcutaneous fat is known as panniculitis. Now let's see some of the terms. Now, abnormal premature keratinization is known as dyskeratosis, and it occurs in squamous cell carcinoma of the skin, Bowen's disease, and Darier's disease. Then, if thickness of the stratum corneum is increased, then it is known as hyperkeratosis, and it is seen in psoriasis and calluses. And if there is retention of the nuclei in the stratum corneum, then it is known as parakeratosis. It is also seen in psoriasis and actinic keratosis. Now, if thickness of the stratum granulosum is increased, then it is known as hypergranulosis, and it is wedge-shaped, and it is seen in lichen planus. Now, epidermal accumulation of edematous fluid in the intercellular spaces is known as spongiosis. It is seen in eczematous dermatitis. There are two types of edema, intracellular and intercellular. This intercellular is known as spongiosis, and if intracellular edema occurs, then it is known as ballooning degeneration, which is seen in herpes simplex virus infection. Now, if there is hyperplasia or increase in this stratum spinosum layer, then it is known as acanthosis, and it is seen in acanthosis nigricans or psoriasis. And if there is separation of epidermal cells, which is because of the loss of the attachment between keratinocytes, then it is known as acantholysis. It is seen in pemphigus vulgaris. Smear To detect this is Lang smear, which is cytodiagnostic test, and cells here are seen as prominent nucleus with perinuclear halo. Now, what are the reasons of this acantholysis? There are three reasons. One is autoimmune, which is seen in pemphigus group. Another is infection due to HSV, which is viral or staphylococcus bacterial or bullous hepaticum. Genetic, which is Hailey-Hailey disease and Terrier's disease. Now let's see desmosomes. These desmosomes are structural support via intermediate filament interactions and autobodies against desmoglin 1 and or 3 causes pemphigus vulgaris. These are desmosomes. And another important here is hemidesmosomes. Hemidesmosomes connects keratin in basal cells to underlying basement membrane. And markers here are K5 and K14. Autoantibodies against hemidesmosomes causes bullous pemphigoid. Now let's see these cells present in the epidermis. There are two types of cells, keratinocytes and non-keratinocytes. Keratinocytes contains 90% of all the epidermis and it is derived from ectoderm. Non-keratinized cells are of three types, Langerhans cells, melanocyte and Merkel cell. Let's see some feature of these cells. Location. Langerhans cells are seen in stratum spinosum, whereas melanocyte and Merkel cells are seen in stratum basale. Embryologically, Langerhans cell is derived from bone marrow, melanocytes are derived from neural crest cells, and Merkel cells are derived from ectoderm. Langerhans cells contain word-break granules, and on electron microscopy, they appear as tennis racket. Melanocyte contains melanosomes, and Merkel cell contains neurosecretory granules. Function of Langerhans cell is like of that of antigen-presenting cells. Melanosomes contain melanin pigment, which is responsible for our skin color, and Merkel cells are responsible for touch sensation via its mechanoreceptors, which are slow adapting and of low threshold. Now, there is one entity called epidermal melanin unit, which is responsible for uniform skin color in our body. One melanocyte contains melanosomes, and one melanosome transfers this color to 36 keratinocytes, and this is known as epidermal melanin unit. Now, let's see some markers. Langerhans cell is detected by S100, CD1A, and CD207. CD207 is Langerhans, which is most specific and seen in Langerhans cell histiocytosis. Melanoma markers are S100, HMB45, and Milan A. And Merkel cell markers are cytokeratin 20. See the location, Langerhans cell, Merkel cell, melanocytes, tennis racket appearance of Langerhans cell in electron microscopy. Thank you.